Dear friends of excellence in science and curiosity-driven science, dear ambassador, ladies and gentlemen, we are going to have a lecture given by a Kavli Prize laureate. The Kavli Prizes are awarded to people for highlighting their outstanding scientific research within 3 fields: astrophysics, nanoscience, and neuroscience. The prize winner, I'm Nils Christian Stenseth, the president of the Norwegian Academy of Science and Letters. And I'm part of 3 institutions that is awarding the Kavli Prizes. This is a partnership between the Norwegian government, who gives funding for organizing the Kavli Prizes, the Kavli Foundation, who gives the prize money, and us, the Norwegian Academy of Science and Letters. And our job is to select the prize winners. And we do that with committees in each of these 3 fields, consisting of 1 from the Norwegian Academy of Science and Letters, and members nominated by learned societies around in the world, nominated by National Academy of Science, Royal Society, Max Planck Foundation, the French Academy of Science, and the Chinese Academy of Science. The prize is meant to highlight excellence in science, which is supported by the Norwegian government. And I'm very happy to have the Norwegian ambassador here to listen to what is being said and to report back to Norway and the Norwegian government. Basic science is very important, because everything we touch upon in our daily life is, as Fred Kavli has said, a result of basic science. And it is increasingly becoming more and more important, basic science, again, as Fred Kavli says. And the Kavli Prizes are, as I've already said, really meant to honor excellence in science. The first one was given in 2008. And here are prize winners in the 3 categories. And in the main street of Oslo, Karl Johan, Champs-Élysées of Oslo, we have banners with Kavli Foundation, Kavli Prizes, which is a fantastic platform for highlighting to the public the importance of curiosity-driven excellence in science. Then in 2010, we had another cohort. And 2012, we had yet another cohort from which today's plenary speaker was part. On the 29th of May this year, I had the great pleasure as the president of the Norwegian Academy of Science and Letters to announce the new 2014 cohort in astrophysics, nanoscience, and neuroscience. Back to today's speaker, Kåre Bergman. We got the prize together with Denk and Anne Graybill for elucidating basic neural mechanism underlying perception and decision. During the lecture, you will be able to send me, through Twitter, questions that can be asked to Kåre afterwards. Please use this address. With these introductory words, it's a great pleasure to welcome Kåre Bergman on the stage. Please, Kåre.
So I'd like to thank Nils Christian for that lovely introduction and thank the ESOF for the invitation to join you here today and to celebrate many of the things that are excellent about European neuroscience. My talk today is entitled Using Fixed Circuits to Build Flexible Behaviors. And I'm introducing this here with a painting of Adam and Eve in the Garden of Eden by Lucas Cranach. It's a painting that's in Vienna. And I'm using this painting, first of all, because it's beautiful, and second, because it illustrates some of the ideas I would like to talk about today about how the nervous system controls behavior. So first of all, when you look at Adam and Eve in this painting, the social bond between them is tremendously evident. When you look at Adam, you think he doesn't even mind if he's going to be thrown out of Eden as long as she will be there with him. And at a mundane level, perhaps this says something about the importance of behavior in our lives and the importance of our emotional and motivational states in driving our behavior. And that's one of the ideas I want to talk about today. The second thing that you can see in this painting are the deer and the lion and the incredible expressiveness that Cranach has illustrated on their faces. And Cranach was saying in his own way that the emotions of animals have something in common with the emotions of humans, that we can recognize in the behaviors of animals some sorts of motivations that are rather like our own. And that is a second theme that I would like to illustrate today. Not to be anthropomorphic, not to say that animals can feel everything that humans can feel, but rather to be zoomorphic, to say that the complex behaviors, feelings, and cognition of humans were built from fundamental principles that were developed in simpler animals. And the third thing about this painting that I would like to point out to you is that above Eve's head is a snake. And snakes look sort of like worms. And for the past 25 years of my scientific career, I have studied the behavior of worms. And so I'm happy, I see worms everywhere, and I'm glad to see it here present in this picture to introduce you to the talk. So about 100 years ago, scientists first began studying the behavior of animals in systematic ways. And the great European neuroethologists, Nick Utenbergen, Konrad Lorenz, and Carl von Frisch in the 1930s developed very thoughtful and precise descriptions of the behaviors of animals. And they noted that there are patterns of behavior that are recognizable within individuals of a species, so that even individuals that have never been exposed to another member of their species can behave appropriately, whether that's a courtship behavior or a mating behavior, or the ability to even organize a very complex behavior like the foraging behaviors of honeybees. So in modern terms, we can recognize these kinds of patterns of innate behaviors in using terms that they would not have had available to them by saying that that must mean that these behaviors are encoded by genes. And this is a remarkable idea, and it seems like a counterintuitive one, because behavior is rapid and flexible and incredibly sensitive to the environment, and genes are fixed and passed on through generations. And so one of the questions that this observation raises is how genes and molecules can lead to the generation of behavior. How are these molecular instructions interpreted through the biology of the animal, through the nervous system, through experience to give rise to different kinds of behavioral outcomes. Indeed, sometimes behaviors are recognizable not only within members of a species, but between species. And it's this idea of commonality and of genes that might have similar roles that I'd like to play out a little bit for you today. So of all the molecules that we can argue might have an effect on behavior, the one for which the case is perhaps clearest and most easily illustrated are for two small molecules called oxytocin and vasopressin. These are short peptides. They're only 9 amino acids long. The structure of one of them is shown here. They were discovered early in the 20th century, oxytocin in the 1930s, in sheep. And they're recognized for their importance both in mammalian reproductive behavior and in mammalian reproductive physiology. So when a female mammal gives birth, oxytocin, released from her hypothalamus in the brain, floods her bloodstream. And it causes her uterus to contract so that the offspring are born. It induces labor. And in fact, this bottle here shows you that in many human females, labor is induced today by injecting oxytocin if it's occurring too slowly. Now, oxytocin also leads to lactation, the milk letdown response. So simultaneous with the physiology of giving birth, it then also controls the physiology of feeding the young. And finally, oxytocin in the mother leads to strong mother-pup bonding, driving a set of behaviors important in the mother for the survival of her offspring. So it drives not only her affectionate and strong relationship to her pups, but for example, also aggression toward other animals. So this one molecule, oxytocin, has multiple roles in maternal physiology and maternal behavior. But oxytocin is not just present in females giving birth. Oxytocin is present in the brains of all mammals at all times. And it's been implicated in other kinds of behaviors as well. Vasopressin is a very similar peptide with similar receptors, and together, oxytocin and vasopressin have been implicated in many mammalian social behaviors. And these include behaviors such as recognition, social recognition of familiar individuals, and aggressive behavior, and mating behavior, and in some rodents, pair bonding behavior. So oxytocin is released during childbirth. Oxytocin is released during orgasm. These are powerful experiences. And so many people have wondered, what would happen if I took some oxytocin? And the answer is, it's surprisingly subtle that people have been administered oxytocin, and it doesn't cause obvious behavioral changes. It doesn't cause very obvious emotions or cognitions. It does cause changes, however, in brain activity as measured by blood flow, and it causes changes that can be measured in certain kinds of carefully designed computer trials. And so there's reason to believe that this molecule has effects on human behavior that we still don't really fully understand. Despite the subtlety of these effects, oxytocin has invaded popular culture to a degree that is ridiculous, perhaps embarrassing. So you can buy oxytocin spray. You can have oxytocin tattooed on your foot. You can buy oxytocin candies, which do not actually contain any oxytocin. I would like to point out to you that oxytocin was first identified in a sheep, and therefore the idea that it is a moral molecule responsible for love and prosperity, as illustrated by that book on the upper right, is that the probability of that is really very low. But what I would like to say is that this hype, which is kind of absurd, is a sign of something that people recognize, which is that people recognize that they have internal motivations that are strong and that are not entirely cognitive, that they might recognize as being some sort of relationship with their underlying biology and their emotions and behaviors. And while I won't tell you anything as interesting as how you might generate instant trust and beam thoughts into the brains of your friends as they're on the lower left, I will try to dissect for you some of a story about how oxytocin and similar molecules can affect the brain to modify behaviors. And here is really the summary of what I'd like you to understand from the talk I'm going to give you today. So when we think about the brain, we have to think about the brain at many different time scales. The brain is something that operates at the level of microseconds or milliseconds to help you generate a thought or a perception or an action. And this kind of property of brain activity is something that's been widely studied in neuroscience, the electrical and chemical transmission of information through the brain that really does happen in milliseconds. This is superimposed on the structure of the brain, which is hardwired, brain structure or connections, which are formed early in development, which are slowly modified over the years by experience, by learning. And these form stable structures that might last days or months or years. But in addition to these hardwiring stable structures, and in addition to this very rapid millisecond level of activity, there's a third way that the brain functions. And this third way I think of as soft wiring. It's a flexible way in which the structures of the brain allow activity to flow in different ways, in different contexts, depending on the needs of the organism. And these are the roles of neuropeptides and neuromodulators, small molecules that function in the nervous system, that can act not at fast chemical synapses locally in the way that classical transmission occurs, but rather over greater distances and over slower times to modify the flow of information over structures as a whole. And these molecules include neuropeptides like oxytocin. Now, these molecules, because they're slow and their effects are subtle, have not been studied as much through the classical methods of neurophysiology and neuroanatomy as the molecules involved in rapid chemical and electrical transmission. But these molecules tell us that they are important, and they tell us they are important in two ways. First of all, these molecules, these modulators, are the subjects of many drugs of abuse and many of the drugs we use to treat psychiatric disorders. So, for example, serotonin, which is used to regulate mood disorder, functions as a neuromodulator through G-protein-coupled receptors. Neuropeptides like opioids and conversely drugs of abuse like peptides regulate the pain system. Mutations in these molecules can also lead to various kinds of complex neurological and psychiatric disorders. And so we believe that these systems, these flexible, soft-wired systems, are important in regulating sleep, hunger, pain, bonding, social behaviors, mood, and addiction. Clearly important roles of behavior, many of which might well be thought of as operating at this intermediate time scale, not at the fastest and not at the slowest. So what we would like to understand is how molecules are interpreted by the brain to give rise to complex thoughts and behaviors. And this is a very challenging problem in the human brain. Your brain has 86 billion neurons. There are more neurons in your brain than there are human beings on the earth. There are more connections between those neurons than there are stars in the Milky Way. It is an incredible structure. It is built, remarkably enough, from only about 25,000 genes. A very small number of components are sufficient to generate this enormous complexity. And it accounts for your tremendous cognitive and emotional abilities. Now, we would like to understand this process, but it is daunting to understand it in a system of this complexity. And so what many neuroscientists do is to study the brains of simpler animals. And I study the brain of a very simple animal, the nematode worm Caenorhabditis elegans. This animal, illustrated there, is only 1 millimeter long. It's the size of a comma on a page of print. And this small animal lives in the wild. It eats bacteria. It can move. It can smell. It can touch. It can learn. It can mate. It can interact with other nematodes. And it does all of these things with a tiny nervous system, a nervous system of just 302 neurons. And that nervous system contains within it about 20,000 genes, of which a huge number are actually very similar to those of humans. And I would just like to point out how surprising that is, even to those of us who study simple animals, that where people have understood genes, for example, in humans, that are responsible for complex human or required for complex human abilities like language or complex human disorders like autism, very often those molecules are also present. Those building blocks are present in simpler brains as well. And among the molecules that we find, both in worms and in humans, are molecules called neuropeptides, these small molecules, including oxytocin. And so we can try to study those molecules in this much simpler animal. So for example, when we look at the genome of C. elegans, we find within it one molecule that's similar to the two human molecules, oxytocin and vasopressin. So things are more complicated in humans. There are two genes, where in worms there are just one. And similarly, when we look for the receptors, the molecules that detect this the oxytocin peptide, in humans there are 4 of them, and in worms there are 2. We can recognize these molecules based on their similarity, and we can show that their relationships to each other are also quite similar between worms and humans. So for example, we can show the biochemistry of this molecule, which is called nematocin in nematodes, will activate the same receptors as are predicted by the similarity to the receptors in humans. So this gives us a sense of biological, biochemical similarity, but in fact, the biochemical relationship between this peptide and its receptors was already known. It had been known for a long time. What we can do in C. elegans that is so much harder to do in a complex brain is to relate that biochemistry to the brain. This is the complete wiring diagram of the C. elegans brain. So from work from John White and his colleagues done in the 1980s, the entire map of all of the connections of all of the nerve cells in the worm brain was assembled. And every nerve cell has its own name, which is shown on this graph. Every nerve cell can be identified in every single worm. We know what genes each nerve cell can make. We know what that nerve cell does. We know what other nerve cells it's connected to. So we have here what's called a connectome, a complete map of how information might be flowing through the fast chemical synapses, the fast chemical and electrical transmission that is so much loved of neurosciences. And so we can take this map, and we can take our molecules, and we can ask how these relate to each other. So for example, we can look at the C. elegans oxytocin homologue, the nematocin molecule, and we can ask, where in the nervous system is this molecule made? And we find that this molecule is made in a couple of places, but one of them that really struck our imagination is that this molecule is made in this slender worm here, which is a male. And it is made specifically in the male C. elegans in a neuron in its tail, a tail structure that is specifically required for mating with hermaphrodites, which are basically females, this larger one below. Now, not only do we find oxytocin in the male's tail, we find that many other neurons within the male's tail make receptors for oxytocin. These neurons are also required and involved in mating behaviors. So we see a potential relationship between oxytocin in the worm and its reproductive behavior. Can we show that this is really true? And also, what kind of changes do we see? We were able to obtain animals that lacked the C. elegans oxytocin or its receptor. And when we found animals that did not have that gene and examined their behavior, we found that those animals survived, they grew up, they did most things fairly normally. They could smell, they could learn, they could move through the environment. But males that lacked nematocin, this oxytocin-like molecule, were not able to mate efficiently. They were only about a third as able to mate as a normal male would be. Now we can look at that mating in detail. And we can look at it because mating in C. elegans, just like mating in other animals, involves a series of different actions over time. It develops in a sequence over time, recognizing a contact with the possible mating partner, and a series of movements around her, turns around her, searches for the right location, and ultimately insemination. We can study all of these very carefully, both in normal males and in males that lack nematocin, oxytocin. And what we see is something quite interesting and surprising. We see that all of the fast actions of mating are okay. The animal can do all of the rapid movements and all of the rapid turns and perceptual events that it tries to do in normal mating behavior. But it does them in an abnormal sequence and with abnormal timing. So if mating is supposed to go here from the left to the right in an ordinary order, we see that sometimes males that lack this will go in reverse order. Or that sometimes they'll get stuck at one step and they'll do it over and over and over again instead of moving on to the next stage of mating. And so what this molecule seems to be doing is not organizing individual actions, the fast actions, but rather the larger overall sequence of a mating behavior. And so we can start to think about the way that these molecules work, relating them to the wiring diagram and to what happens when they're removed, as a way of thinking about mating behavior is really involving two different pathways in the brain. It involves fast pathways, shown here in black, where different neurons detect different things about a mating partner in the environment and generate fast actions like turning and searching. But it also involves a slower pathway determined by the neuropeptide nematocin, which modifies the action of those fast pathways in a mating context, tells the animal it's in a proper mating context, and modifies the flow of information accordingly. And just to give one very simple example of a way in which it modifies that, if you touch a male lightly when it's not mating, it avoids that touch. But a male that's touched lightly when it is mating finds that touch appealing, and it's a sign of continuation. That switch between those two behaviors is one of the things that oxytocin is trying to do, changing the animal's response to a touch to be appropriate to the mating context, rewiring the behavior. So to summarize this part of my talk, we can think of genes as something like a vocabulary for biology. If we have 20,000 genes in a worm or 25,000 genes in a human, we might think of those as words that can be used to build a simple language, the simple language of the worm brain, or a very complex language, the language of our own brains. But we can think of the genes as having some sort of a function or meaning that might be similar in different languages. And in fact, our results, taken together with other results across the field in other animals, suggest that this one gene, this one molecule, oxytocin, seems to have a role in reproductive behavior across a huge variety of animals. And these include mammals, such as rodents. They include our nematode worms, and they include other animals from very different parts of the evolutionary tree. For example, these leeches studied by Bill Kristen's lab. And finding this in so many different branches suggests very strongly that very early in evolution, these molecules became allied to reproductive behavior, and that they've been organizing behavior in some way like that over a very long time. So oxytocin represents one extreme example, and mating represents an extreme example of something that's actually a more general function, which is that behaviors are not just something that happens from instant to instant, but something that is organized over time. And that in fact, in the brain in general, behaviors need to be organized. Groups of actions need to be organized together in organized, coherent behaviors. And we think that these may be the places that slow transmitters, the neuropeptides and neuromodulators, are particularly important in brain function. Now, the question that we want to ask in more detail, and that we can ask better by studying more examples rather than always focusing on a single example, is what do we see when we look at different neuropeptides or different neuromodulators and different behaviors? What can we see that is in common between them? Can we start to extract some principles, some general rules of how behaviors are generated by studying multiple systems? So for example, we know that sometimes neuromodulators are released very locally at synapses. We know that sometimes they're released from deep within the brain, very broadly within the brain, or even through the whole body like oxytocin. But how general are these? Really, what is the structure of the way that a slow transmitter modifies brain function? Are they really just like another transmitter, but a little more subtle? Or are they really very broad? The word that's sometimes used to describe this are sprinkler systems that are just bathing the worm in transmitters. Or are they something in between? And that's the question that we really wanted to ask ourselves. What is the structure of these circuits? What distances do they act over? What time scales do they function over? And how do they relate to those fast synaptic circuits whose structures and fast electrical activity are so broadly studied in neuroscience? And the next story I'll tell you about is about a different kind of behavior that has quite different from mating behavior in a certain number of ways. It's a natural behavior that all animals show all the time, but it has some other similarities as well. And the behaviors that I'd like to talk about are just the general behaviors that animals show when they're moving around freely and spontaneously and organizing their own behavior. And what happens when, under those circumstances, is that animals spontaneously organize their behavior into structures, long-term structures, where they're active for some periods of time and inactive for some periods of time. Now, the most extreme version of that is shown in this slide up here at top. And this is a slide I took on purpose. It's from the first real paper that related genes to behavior, a paper by Seymour Benzer showing that there were genes that controlled circadian rhythm in flies. And this just shows activity tracks of flies, and what you can see here is that flies are very active during the day, and they're inactive during the night. They sleep, and they're awake. And so this is an example of a spontaneously generated, biologically regulated, long-term structure of behavior, sleeping and waking. But even within waking states, animals tend to spontaneously regulate their behavior. So when animals are awake, they'll move around very actively some of the time, and they'll be quieter some of the rest of the time. And that is a phenomenon that is seen in all animals. It's seen in the flies in the lab. It's seen here. I've taken an example from a paper by Carl Dijsselroth, who gave one of the keynote talks earlier today, of animals showing swimming behaviors that are used to assess different kinds of emotional responses in rodents, where the animal will swim fiercely for some period of time and then just float in the water some of the time, but it's still awake. And it's seen in wild animals that are foraging in the wild. So if you see birds flying around or deer grazing, they will stay in one area for a small time, apparently quietly, and then they'll very actively move to a different location. So this is an overall structure of behavior that can take place over minutes, over hours, or over a day. And we started to study this behavior in our tiny worm, C. elegans, because of work that had been done by Steve McIntyre's lab, showing that when C. elegans is moving spontaneously on food with no obvious outside stimuli, it will spontaneously alternate between different kinds of behavior. So what it will do is it will sit quietly sometimes on the food, moving back and forth rather slowly and eating the food in a small area. And then spontaneously, without any apparent external stimulus, it will suddenly start moving to a very different part of the food. And it will do that for a while, and then it will switch back. And these two behaviors are called dwelling and roaming. And I'll show you a movie of that here in a slide. There'll be an animal that starts dwelling in this part of the slide here. And you can see it moving in a small area, turning rapidly, eating a lot of the food, and then suddenly it roams. That was roam. Now it's dwelling again, roaming, dwelling, roaming, dwelling. This movie is sped up 100 times. That makes worm movies much more exciting. And so what you can see is that the animals seem to be just sort of switching back and forth between these different behavior patterns. And in characterizing this behavior, several groups studied this and showed that they showed the properties of these kinds of internally generated spontaneous behavior. And they're not completely independent of the environment. So they seem to be regulated by how good conditions are overall. So when animals are under good conditions, when they're happy with where they are, they spend most of their time dwelling. But when they're a little bit dissatisfied, for example, if the food is low in quality, or if there isn't quite enough food, or if there are environmental irritants that are bothering them, then they spend much more of their time roaming. So the idea is that dwelling is an exploitive behavior where we exploit a desirable environment, and roaming is a behavior where we explore the environment to see if there might be something a little better going on. So they're both internal states and external cues that regulate these behaviors. So Steve Flavell, a postdoc in my lab, decided to try to take a molecular approach to understanding what roaming and dwelling were, and to try to understand how these behaviors are generated. And I'm just going to tell you sort of briefly how we go about analyzing behavior in an animal, just to point out that we don't just sort of watch movies, that the importance of studying behavior in many cases is to really be quantitative and rigorous and analytical about what you measure. So we take many hundreds of hours of videotapes, like the videotape I showed you in the past, and then we track the animals through a computer, and we examine different features of the animal's movement. And so, for example, we can measure two features of the animal's movement very easily, one of which is how often does that animal change direction, and the other is how fast are they moving. And what you can see is that animals go through periods where they change direction a lot, but move rather slowly, and they go through periods where they move quite quickly and change direction infrequently. And these overall structures are correlated. They're not randomly distributed in time. And these suggest to us that there's an underlying behavioral state, an active state and an inactive state, this roaming state and this dwelling state. And we can characterize those quantitatively in different ways. We can put numbers on them to describe what they're doing. So for example, we can show that dwelling states are, in general, 8 minutes long, a pretty long time period. Roaming states actually fall into two classes. One kind of roaming event is 1 minute long, and then there's a second kind of roaming state that's 9 minutes long. These states aren't cyclical. It's not like sleep and waking on a circadian rhythm. They seem to occur randomly. So trying to understand what the structure is and what the timing is remains an open question. That was the next step of what Steve decided to do. So what he did was to undertake a genetic examination of what it was that determined roaming and dwelling states. Now, each of these states involves animals moving around. Each of them involves feeding and laying eggs and changing directions. It's just the distributions of those events over these long times that are different. And he was particularly interested in these long times. What is it that makes a behavior that lasts for 8 or 9 minutes? How do these long-lasting behavioral states take place? And what he discovered from genetic analysis is 2 major pathways that are antagonistic to each other, each of which seems to be primarily involved in defining one of these long-lasting behavioral states. One of these molecules is serotonin. Serotonin is a highly conserved molecule. It's found in our brains, it's found in worm brains, and it's one of these neuromodulators. It's the target, for example, of most of the major antidepressants that are used in humans. And I'll tell you about how serotonin seems to be important in the dwelling or quiet exploitation behavior. The second molecule is a neuropeptide. Its name in worms is PDF. It's related to a molecule in mammals called VIP, and it has the exact opposite effect. It seems to be important in the more active or exploratory roaming states. So what do we see about these molecules? Here are behavioral tracks and comparisons of animals that are normal or wild type, or in animals that are missing a receptor for serotonin. It's an inhibitory receptor, a receptor that when serotonin is present, silences the neuron that expresses it. And we would see similar things if we were looking at animals that lacked all serotonin, because we also have mutants that cannot make this neurotransmitter in C. elegans. And what we see is if we compare these animals to wild-type animals, I'm going to use the same color code in all slides. Red is the active roaming state, and blue is the calm exploratory dwelling state. Under particular circumstances, wild-type animals dwell about 80% of the time, whereas animals lacking serotonin only dwell about 40% of the time. They're much more active. But when we look at our careful behavioral tracks and we look at the individual structures of the roaming and dwelling behavior, they're normal. When an animal is dwelling, its dwelling looks perfectly normal. It just spends much less of its time dwelling than it should, and much more time roaming. And we can see that if we look, for example, at the duration of a dwelling event. Now, instead of being 8 minutes long, they're only about 3 minutes long on average, and roaming durations have become, on average, much longer. So the same idea of a normal behavior with really abnormal timing shows up in exactly the opposite way in animals that lack the PDF peptide. These animals dwell all the time. This is a 90-minute movie of an animal dwelling. This animal, instead of switching back and forth between roaming and dwelling states, is simply occupying a dwelling state. Now, very, very rarely, these animals will tear off and start a roam, about 5% of the time. And when they do that, that roaming state is almost normal. And so again, what we seem to have here is something that affects the duration of a behavioral state, not the state itself. And again, this happens by changing the length of the states. Dwelling states have now become much longer than normal, and roaming states much shorter. So this gives us relationships between some kind of behavioral state and molecules. And I should say, we view these behavioral states as models for something like motivational states or emotional states. We do not think of these as any particular emotion. We do not think that worm serotonin has anything to do with worms being depressed. What we think is that these molecules are modifying the way the nervous system works, and that we can use them to understand how molecules like this operate on groups of neurons to modify behavior. So they're a general analogy we believe holds up, the idea of molecules modifying behavior, but we don't try to make a one-for-one representation of a worm behavior to a human or even mammalian behavior. But we can certainly learn more than just this general idea that serotonin has something to do with long-lasting states, because we have that wonderful worm wiring diagram to work with. And I should say that serotonin had long been known to exist in C. elegans. And one of the remarkable things about working on C. elegans is that there are exactly 11 neurons in C. elegans that make serotonin. And they fall into 6 classes. And they're distributed in different parts of the body, including the mouth, and in sensory neurons and in motor neurons. And we can ask a very precise question. We can say, which neurons are important? What's important about serotonin? Is it the total amount of serotonin that's important? Is it important that it be made in one specific neuron, or the average amount of neurons, or any combination of the neurons? What is it that defines the relationship between this molecule and this behavior? And we can do that using genetic techniques that allow us to eliminate serotonin specifically in each of those individual neurons while leaving it present in all of the other neurons, and systematically test animals that are just missing serotonin from one kind of neuron and asking whether they can perform normally in behavior. And again, these are behaviors that we do quantitatively, where we compare animals that have been knocked out for different sorts of neurons for serotonin, and we ask how those behaviors behave in these sorts of long quantitative tracks. And what we find is that exactly 2 neurons are important out of those 6 classes in regulating roaming and dwelling. The 2 neurons, which have worm names, which we can name and point to in every worm, NSM and HSM, are required for the normal regulation of roaming and dwelling. And we can do exactly the same experiment with the serotonin receptor. There are many worm neurons that detect serotonin. There are actually multiple serotonin receptors, but only one receptor is important, and we can look at, of the neurons that make it, where it must be present, and we find that some, but not all, of the neurons that make this receptor are important in the behavior. We see specific rules about how the molecule and the receptor are made. And these rules can be conceptualized in this little diagram here. There are 2 neurons that make serotonin, shown in blue. And those 2 neurons, shown in blue, the cool color, promote dwelling. They inhibit, through their receptor, MOD1, 3 other neurons, shown in red. Those 3 neurons, when they are active, promote the active roaming behavior. When they are silenced by serotonin, roaming behavior is suppressed. Now, these are random names, and if you're not a C. elegans researcher, their names don't mean much to you. But when we look at them, we can relate them to everything else we know about the worm's brain and about the worm's wiring. And we can really start to think about what this means. So, for example, the first thing we see is that none of these neurons are connected to each other in the anatomical wiring diagram of C. elegans. All of these neurons have synapses with other neurons, but the serotonin synapses are completely independent of the fast chemical and electrical synapses that are used to regulate fast actions. It's a second wiring diagram on top of the first one. So what's the next question we'd like to ask? How closely does serotonin relate to these animals' behaviors? When are the neurons active compared to when animals are roaming and dwelling? Here's a question that we ask using genetic tools that have been developed in recent years that enable us to look at the activity of single neurons by fluorescence traces in those neurons. So this asterisk marks a single spot in the worm's head. We can label just the NSM neuron in the worm's head and ask, when is NSM, which makes serotonin, active? When it becomes brightly fluorescent? And what is the worm doing when NSM is active? We can let the worm move freely, monitor that activity, quantify it, and then look at the traces of when the neuron is active as it moves, and see, for example, here, a little spike of activity in the neuron associated with an animal's reorientation at a particular time. So we can take many hours of these kinds of movies and then relate them together and ask, when NSM is active, what does a worm do? And the answer is, when NSM becomes active, it generates peaks of activity, and associated with those are decreases in the worm's speed. And decreases in the worm's speed are part of dwelling behavior. The peaks of NSM activity coincide with dwelling. Conversely, when NSM activity falls, animals begin to roam. So the activity of the serotonin system is related to the natural transitions between roaming and dwelling. And here's something pretty interesting. Most neurons in C. elegans, like most neurons in your brain, are active very rapidly and turn off very rapidly in seconds or fractions of a second. But this neuron that makes serotonin is active for like a minute or a minute and a half at a time. It's a slow neuron. And that's really interesting because we're interested in a slow behavior, slow roaming and dwelling behavior. So this is a relationship. Can we prove that serotonin is important in this behavior? The way that we can do that is to make use of other new genetic tools developed by a number of winners of the European Brain Prize from 2 years ago, including Carl Dijsselroth, who's here, which allow you to perturb neural functions and to imitate or to play in the natural patterns of activity you see using artificial genetic methods. We can activate a neuron of our choosing using genetics and a molecule called channel rhodopsin to activate the neuron. We can inhibit a neuron of our choosing at a time of our choosing using light and a molecule called archirhodopsin in a transgenic approach. And so we can, using these, play back to the worms the patterns of activity in the serotonin system that it normally shows. We can imitate to the worm the normal pattern of serotonin activity, which would happen if you turn these neurons on or turn these neurons off. And if we do that to animals that are roaming, and we imitate serotonin, we convert them into dwelling behavior. We can basically play into the nervous system the serotonin signal and elicit the dwelling behavior. And conversely, if we antagonize the normal functions in animals that are dwelling, we can cause the animals to start roaming, either by shutting off their serotonin neurons or by activating those targets. So these molecules and this little circuit actually appear sufficient to drive the transitions between these two behaviors. Not only do they drive the two behaviors, they drive the two behaviors here, as you can see clearly for roaming, in a way that persists after the signal is gone. They're driving long-lasting behaviors. And perhaps my favorite experiment here is an experiment where we can use these activation methods to actually prolong a pre-existing dwelling state. We can take animals that are already dwelling, and then we can give them a little pulse of serotonin during the dwelling state. And what we find is that they continue to dwell, no big change there, but that dwell is prolonged. That 1 minute of serotonin makes the animal dwell for 6 minutes longer than it would have dwelled otherwise. And we're now incredibly close to the normal time scale of roaming and dwelling behavior, with the animal 8 or 9 minutes, where we're able to start to see that the natural pattern of activity, a minute of activity, converts into a natural behavior 6 minutes long, very similar to what we see spontaneously. So Steve has done the same experiments with the opposite side, the roaming side or the active side. And the results are really remarkably similar. Both PDF, the peptide for roaming, and PDF receptor, its receptor, are required in multiple neurons, in some but not all of the neurons that normally express these molecules. And he can also do optogenetic experiments to show that these molecules are sufficient to induce roaming behavior. He had to use different genetic tricks to do this because PDF does not directly activate or inhibit neurons. It acts indirectly to increase the amount of a cyclic AMP second messenger. He has to use light-activated molecules that will create that messenger. But if he does so, he can very effectively convert animals from a dwelling into a roaming state. So we have two molecules, two systems, one for roaming, one for dwelling. How do they interact with each other? What do these antagonistic systems do? We can eliminate from worms both the roaming promoting molecule and the dwelling promoting molecule and have animals that have neither. What happens when we take both away? These animals still roam and they still dwell, but both of these states are now destabilized. They can only roam for a minute or so, they can only dwell for a minute or so, and they switch very rapidly back and forth between these unstable states. And so that really makes us think the role of these molecules is to prolong behavioral states. Even without both of them, the behavioral states are present, but these are required to take a short behavioral state and turn it into a long behavioral state. And so these are the conclusions of this set of experiments. From our experiments, we've identified neurons required for roaming and dwelling foraging behaviors. We've identified a set of neurons that induce and extend dwelling states and a set of neurons that induce and extend roaming states. Now, these two neurons barely overlap with each other. Only one of the neurons out of the 10 neurons identified in these studies was the same for serotonin and for PDF. So the information is distributed among 10 different cell types out of just 300 cells in the worm. And remarkably, these cell types are different from each other, and they're entirely different from the fast wiring diagram. These neurons are not interconnected at either the PDF or the serotonin site. So what we have here is what we think of as a wireless diagram, a chemical diagram superimposed on the fast circuits that's reshaping the circuits. And we can look at how that relates to where the neurons are and where their synapses are, and we can say it's not completely global. It does seem to be a local action within a short distance, typically a few microns, but it's not synaptic. It doesn't overlap with the wiring diagram, and it's acting at a slow time scale, a time scale of minutes. So how might these principles apply to other nervous systems? Can we take our simple ideas of how modulators are functioning in worms? I told you about oxytocin, about serotonin, about PDF. Can we relate those to what we know about these molecules in other systems? Well, one of the things that's really interesting to us is that when we look into what neuropeptides and neuromodulators do in the mammalian nervous system, we find that they're also required for the stability and switching in behavioral states. And a great example is a small neuropeptide called hypocretin or orexin. It's made by just 20,000 neurons within your brain. But when those neurons are damaged or destroyed, it leads to a very striking neurological defect called narcolepsy or cataplexy, where the sleep state starts to invade the waking state and you don't have a stable waking state. You can suddenly fall asleep or collapse while awake, or even have dreams while you're awake, due to the loss of this stabilizing peptide. Similarly, the neuromodulator dopamine, which is very important in Parkinson's disease, seems to be important in the initiation and the inhibition of different actions. Once an action is started, it's okay. It's really this initiation or triggering step that's dopamine dependent. And when we look at the roles of these molecules, again, I told you at the beginning, we think that neuropeptides and neuromodulators are involved in motivation and emotion. And these are often things that occur over slow time scales. Sleep, hunger, thirst, mood, arousal, social drive, mating, and stress are all relatively long-lasting kinds of responses. So the time course may be part of what's really important about these molecules. I think if there's a lesson that comes out of what we've said, it's that the nature of the anatomical pathways should be mapped more strongly for all of these molecules. When we treat someone with Parkinson's disease, we give them levodopa we give them as a pill, and it affects their entire brain. Now, our work suggests that it's not just that these modulators are acting everywhere, that they have local actions that are important. And so we might think if you map the pathways more carefully, you might get better results. And just to conclude, we think that there are many maps, and that when we look at other circuits, you can think about them being rewired with neuromodulators. We can think about motor circuits, such as those that are affected by Parkinson's disease, feeding circuits that are regulated by hunger and satiety in many animals, even sensory circuits. For example, your own light here in this dark auditorium. Your eye is working in a different sensory modality than it will be working when it goes into the bright light outside. And that switch is made in part by dopamine coming and switching off a set of circuits that are important in the dark but not in bright light. So in conclusion, I've told you about just a few modulators in a small brain. The human brain has many, many overlapping circuits, many more than the worm brain. And we note that they have many neuropeptides and neuromodulators. There are about 100 neuropeptides in the human brain, and only about 20 of those are understood at all. There are enormous reasons for thinking that these things are important. There are clinical implications for how they might be functioning. I think there's a huge amount to think about here in ways that the nervous system functions flexibly and sensibly in different contexts to give you different behavioral outputs. And that neuromodulators are important in selecting among the options presented by a wiring diagram for ways that information can flow through the brain. And with that, I will thank you for your attention and just show you the picture of my group at the Rockefeller University. The work I told you about today was here. Jennifer Garrison did the work on oxytocin, on nematocin, and Steve Flavelle here in the back did the work on serotonin and PDF and foraging behavior. Many other students in the lab developed some of the reagents or the approaches that were used in these experiments. Thank you.
Thank you very much, Corey, for a wonderful talk, showing excellence in science. Good science is to simplify, which you have shown in a fantastic way. There's a question from the audience here, maybe one being a little bit skeptical about some of this. The question is, when, if ever, is C. elegans exhausted in terms of usefulness? And the person adds, and where do you think we should look next?
I think my theory about C. elegans is that you should use it to study things that you cannot study in more complex animals. That the value of this system is to address problems which cannot be addressed in the more complex systems. So at this point, we think that there are a lot of questions within the brain that do need to be studied in simpler brains. We hope, of course, to ultimately understand the brains of more complex animals, including ourselves. But we need to understand something about what the ground rules are to reach that point. So where do you go next? There are different approaches to systems. I think that there are many examples in neuroscience of using different animals to study different kinds of processes, of using the diversity of biology to shed light on different processes of interest. So we know that many scientists study the mouse as sort of a workhorse of modern biology, but one of my favorite areas of neuroscience is the study of bird song. Birds are the only animals other than humans that communicate through acoustic mechanisms, through song, and they learn their song from an instructor. And it's been an incredible system for learning how instruction occurs, how the brain learns, how fast sounds are generated. And so I would say that different questions will show themselves up in different systems, that there will be things to study in birds, things to study in mice, things to study in fish, and in other animals that we haven't even imagined.
I think it's fascinating. I have a question and reflection myself. You have shown very elegantly in this presentation and in your work the neurological base for behavior. And I'm an evolutionary biologist, and we evolutionary biologists, you are an evolutionary biologist yourself, would like to ask, in addition to how things are organized, why they are like that. How can this work that you have presented to us help us understand why, understand the evolutionary questions for why we are behaving the way we are, or why C. elegans is behaving the way C. elegans is behaving?
So I think all geneticists are evolutionary biologists, whether they realize it or not.
I agree.
Because the genomes are records. The genomes are both a set of instructions as to how to solve a problem and records of how the problem has been solved in the past. And so when you look at how genes are interacting with some piece of biology, very often you are viewing both its current function and the historical contingencies that led it to where it is. I think the cases, I'm actually infatuated in the way that scientists are with the subject that I study. And so I think that these neuropeptides and neuromodulators represent very clever ways for the nervous system to modify behaviors evolutionarily. And this is a testable hypothesis. Mm-hmm. So if you want to build a completely new system for fast actions, that requires building connections between two brain areas, making them work effectively. If you want to connect two areas with a neuromodulator, that doesn't require anatomical specialization. That just requires a small change in the expression of a peptide or a receptor. So you could very easily, with small numbers of changes in gene expression, lead to new kinds of behavioral connections. Now, this has been suggested to be true actually with the oxytocin vasopressin system and the social behavior of rodents, and in particular in the relatively strongly pair-bonded species versus much more sort of solitary species of rodents through changes in gene expression in these pathways. So I think that's one way of thinking. But you might still say, that's just another way of answering how. That's not a way of answering why.
But it is on
right?
the way to asking and answering the why question, I think. Thank you very much. Yesterday, at this very stage, there was a discussion whether the previous century was the century of physics, and this century being the century of biology. I should not involve myself in discussing that, but I do think to be a biologist today is fantastic because we can do things, we can ask questions to nature and actually get answers. You have shown a fantastic example why it's fantastic to be a biologist today. Thank you very much, Corey.